AP Biology, this is our lecture on membrane structure and function. Basically, what you need to know from this chapter is why our membranes are selectively permeable, the role of phospholipid proteins and carbohydrates in membranes, how water will move if a cell is placed in an isotonic, hypertonic, or hypotonic osmotic condition, and how electrochemical gradients are formed within the cell. So we know in general that the cell membrane itself, or the plasma membrane, is a selectively permeable membrane in that it allows substances to cross more easily than others. So some substances can readily pass through the membrane from one side of the cell to the other, while others cannot. The cell membrane itself, due to its composition, is what we call now the fluid mosaic model. The fluid mosaic model is widely accepted in that it describes two components of the cell membrane, or two characteristics of the cell membrane, known as the cell membrane being a fluid, in which a membrane is held together by weak interactions and moves. It's not a static membrane. The membrane does move like any fluid would. And when we think of fluids, we think of substances like gases or liquids. It's also a mosaic in that it is composed of many different things. Of course, the major component of any biological membrane is that of phospholipids, but also embedded in the membrane are proteins. You also have carbohydrates that are also found embedded within the membrane. Early membrane models, in 1935, Davison and Dinelli proposed a sandwich model in which the phospholipid bilayer was sandwiched between two protein layers. The problem with their model was that there are varying chemical compositions of membranes. Hydrophobic protein parts are also there. So what we have seen now is what we call today the fluid mosaic model. So using the process of freeze fracture, it revealed the structure of the membrane's interior. And what we know is inside the membrane, you do have the hydrophobic tails. You also have proteins that will span across the membrane itself. As you can see here, you have the hydrophobic regions and the hydrophilic regions of both phospholipids. But if you look, you could also see that on proteins as well. Proteins have both hydrophobic and hydrophilic regions. Of course, the hydrophobic regions are going to be embedded within the membrane, and the hydrophilic regions are going to be peripheral, or to the outside of those channel proteins. The phospholipid bilayer then, when we think of phospholipids having both hydrophilic and hydrophobic components, like their head and tail, we say it's amphipathic. Amphipathic is just as it states, polar and non-polar. The phospholipids form a bilayer, two layers, and ultimately what's going to happen is the hydrophilic layers are going to face aqueous environments, or those environments containing water, and the hydrophobic tails are going to be hidden in between. That hydrophobic barrier is going to keep hydrophilic molecules out. The membrane's fluidity is ultimately affected by low temps. Phospholipids with unsaturated tails is going to cause kinks that prevent close packing. You could see that the difference between in the pictures here, unsaturated and saturated hydrocarbon tails. When you have the unsaturated hydrocarbon tails, they contain carbon-carbon double bonds, and therefore you get kinks in the hydrocarbon chain. Ultimately, this is more fluid than for, say, a saturated hydrocarbon, saturated hydrocarbon tails in the phospholipid bilayer. That's going to be more viscous. And remember, viscosity is a resistance to flowing. Cholesterol is another big factor. Cholesterol resists change by limiting the fluidity at high temperatures, and it also hinders close packing at low temperatures. So you could see cholesterol within the animal cell membrane here. would limit or stabilize that cell membrane. Adaptations, bacteria in hot springs, unusual lipids are found. Also, winter wheat has an increase in unsaturated phospholipids in their cell membranes as well. So you have two types of membrane proteins. You have those that are integral proteins and those that are peripheral proteins. Integral proteins are going to be embedded in the membrane. They're going to be determined, these are determined by freeze fracture. These are going to be the transmembrane with hydrophilic heads and tails are hydrophobic middles. So when you look at those in a minute, you'll see how they have hydrophilic regions and hydrophobic regions. Peripheral proteins are extracellular or cytoplasmic. Extracellular means that peripheral protein would be found on the outside. Cytoplasmic will be found on the inside of the membrane. They are not embedded in the membrane as integral proteins are, but they are held in place by the cytoskeleton or the extracellular matrix of the cell, depending on whether it's extracellular or cytoplasmic. It provides stronger framework. If you look, so here you have the peripheral protein. Here is an integral protein. Also, you can see cholesterol embedded in the membrane there itself. So let's look at specific examples of both peripheral and integral proteins. A transmembrane protein, transmembrane proteins, here's where you could see the N-terminus and the C-terminus region. These two areas here are going to be hydrophilic, and the center there is going to be a hydrophobic interior. So you could see the hydrophilic and hydrophobic ends to this transmembrane protein. And you know, the functions of many of these proteins would be transport. They can serve in enzymatic activity for help speeding up chemical reactions. They'll play a role in a single transduction pathway. So we have single transduction proteins. They'll play a role in cell-to-cell recognition. They'll also play a role in intercellular joining. And lastly, attachment to the cytoskeleton and the extracellular matrix. So you can see that in here. So here are a list of various types of proteins. These are in your textbook. You should have a generalized understanding of these proteins. Those that are channel proteins, carrier proteins, cell recognition proteins, junction proteins, enzymatic, and receptor. There are descriptions there to help you understand a basic example for each. Carbohydrates on the cell membrane are going to function in cell-to-cell recognition, often found in developing organisms. You have both glycolipids and glycoproteins. So if the carbohydrate chain is attached to a fat, we call that a glycolipid. And if it's attached to a protein, like a channel protein, we'd call that a glycoprotein. An example would be blood transfusions are type specific. So when you're doing cell-to-cell recognition, that's where something like a carbohydrate would play a role. So here you can see the synthesis of things being manufactured in the cell. Here you have the endoplasmic reticulum, and you have the inside of the endoplasmic reticulum, which any space is referred to as the lumen. You have a transmembrane protein expanding across there. So this vesicle is going to transport ultimately to the Golgi apparatus, and in it, it's going to contain, in that transport protein, you're going to have a secretory protein. You also have things in there for recognition, like a membrane-bound glycolipid for attachment there. So ultimately, this will come up to the Golgi apparatus where it's going to be modified and packaged, and then what will happen is you'll have a vesicle secrete off then, and this would be a secretory vesicle, where that vesicle is going to be transported up to the cell membrane of the cell. And ultimately, what will happen is that vesicle is going to bind to the cell membrane of the cell and expel or secrete that protein to the outside. So selective permeability. Small molecules, both polar or nonpolar, cross easily hydrocarbon, hydrophobic molecules, carbon dioxide, and oxygen. Hydrophobic core prevent passage of ions and larger polar molecules within the cell membrane. So when we look at the different types of transport, we're going to look at both type of transport that does not require energy and a type of transport that does require energy. Passive transport is a type of transport that does not require any input of energy. So no energy in the form of ATP is needed. This would be diffusion, and we say that in diffusion, you're going down the concentration gradient. It's a downhill movement when we think of going from high to low. So molecules are going to go from areas where they're highly concentrated and move in the areas where they have where they are at a lower concentration. Examples of things that would experience diffusion are hydrocarbons, carbon dioxide, oxygen gas, and water, although water is given a special type of transport. So if you look here, you have a membrane and you have molecules of dye, and ultimately you could see that on one side of the membrane the dye is highly concentrated, and on the other side the dye is at a lesser concentration. So ultimately you're going to have a net movement or net diffusion from one side of the membrane to the other. That net diffusion will continue until the concentration of dye molecules is at equal concentrations on both sides of that biological membrane or membrane in general. And at that point, you would say that you have reached equilibrium. So if you look at a scenario then with two solutes or different molecules, you could see how the net diffusion would take place until equilibrium is established between both the purple and the orange spheres there to represent solutes across the membrane. So when we think of concentration gradient, you're looking at the concentration of a solute on one side of the membrane compared to the other, and the gradient is going to be that area in which it travels. So the space in which it travels will be from one side of the membrane to the other. Osmosis is a special type of diffusion. It is the diffusion of water across a membrane. You can see that here looking at the osmometer. The osmometer is, let me get back to the slide here. This would be representative of an osmometer, and there you could see a selectively permeable membrane in there. So you have a low concentration of solute sugar on one side of the membrane compared to a higher concentration of the sugar solute on the other side of the membrane. Ultimately, now what will happen is water molecules could diffuse across the membrane. So water is going to travel in the direction from this side over to the other side where the sugar molecules are highly concentrated. Because in the area where the sugar molecules are highly concentrated, you have a lesser concentration of water. So osmosis is the diffusion of water molecules. And we could see that in our picture here. We could see that external environments in the cell can ultimately be hypertonic, hypotonic, or isotonic. And you can see the various examples here for both the animal and the plant cell. So in a hypotonic condition, an animal cell would be lysed. Lysis is to burst or to rupture, ultimately because that animal cell lacks any cell wall. But if with the plant cell would take on water, and ultimately we would say it is turgid. Remember that the plant cell, the water is stored in the central vacuole, and the vacuole is surrounded by a membrane called the tonoplast, and the pressure inside that central vacuole is called turgor pressure. So that pressure would be exerted outward on all parts of the cell and would basically make that cell turgid. And that cell would not erupt due to a cell wall. In an isotonic solution, isotonic solution is when you're at equilibrium. In the animal cell, it's normal. In the plant cell, it is flaccid. And then in the hypertonic solution, the animal cell would be losing water and ultimately it would shrivel. In a plant cell, the plant cell would lose water in a hypertonic solution, and we would say the condition there is plasmolyzed or plasmolysis. So our whole lab is going to focus on understanding osmosis and diffusion, and in there we need to understand what water potential is. And ultimately water potential, or psi, is when we talk about how water is going to move from an area of high potential into an area of low potential. So the water potential equation, ultimately water potential, or psi, is going to equal the free energy of water. And then you have the potential of the solute concentration, or osmotic potential. And then you have the pressure potential, or psi p, which is the physical pressure on the solution. In the case of plants, you would consider that turgor pressure. So these are the equations. This is the equation for water potential. So you have psi equals the solute potential plus the pressure potential. We know that pure water itself has a pressure potential of 0 molar pascals. In plant cells, it is 1. So if we look at calculating the solute potential using that given equation, the solute potential equals -1 CRT, -1 I CRT, not -1, -I CRT, sorry about that. So the solute potential, again, is equal to the ionization constant, and the ionization constant is going to be the number of particles made in water. And ultimately, you also have capital C, which is the molar concentration of the solution, and you have R, which is that pressure constant at 0.0831 liter bars per mole kelvins. And then the temperature, of course, would be in kelvins itself. So it would be, remember to determine kelvins, it's degrees Celsius plus 273. So the addition of solute to water lowers the solute potential and therefore causes it to become more negative, and therefore will decrease the water potential. So that would be if you were to add a salt or a sugar. So where will water move? Water is going to move from an area of high potential into an area of low potential, where you have a more negative potential. The solute concentration, if you look at it, the solute concentration is going to go, you'll see water will move from an area of low solute concentration into an area where it's the solute concentration is much higher. This is because in that higher solute concentration, water is, of course, going to be at a lower concentration than what it is if it were in a low solute concentration. So here you can see movement of water across a membrane. You have dilute sugar, water. Water is more concentrated here. And then you have concentrated sugar solution on this side of the membrane. So ultimately what will happen is water is going to move from one side of the membrane to the other. So it's going to move from an area of high pressure into an area where it's at low pressure. So these are all the factors that will cause water to move. So if you look here, here are some examples. Which chamber has a lower water potential? Which chamber has the lower solute potential? And in which direction will osmosis occur? So if one chamber has a potential of -2,000 kilopascals and the other is -1,000 kilopascals, which is the chamber that has the higher potential? We'll look at some of these scenarios and do calculations in class. So low water potential atmosphere, the leaf, when we think of the low water potential of the atmosphere, the potential is about negative 95.2 molar pascals. Of course, that would change with humidity. Changes with humidity usually are very low. The leaf potential is about negative 0.8 molar pascals. It depends on the rate of transpiration. Low when the stomates are open. And we'll look at that a little bit when we get into photosynthesis. So we'll do sample calculations in class. Facilitated diffusion is a type of diffusion in which you are going to need a transport protein, whether it be a channel protein or a carrier protein, to help hydrophilic substances get across the membrane. It is a type of passive transport because you are still moving molecules across the membrane from areas of where they are highly concentrated and moving them into areas of where they are lowly concentrated. This can be done two ways. You can provide a hydrophilic channel or loosely bind a carrier molecule across. So you can see what a carrier protein will look like versus a channel protein. And we'll look at examples of this as well. So examples of this would include ions, polar molecules like water, and glucose. We now know that aquaporins are channel proteins that are going to allow for the passage of water across the membrane. And here you can see what they look like structurally. If you look at these diagrams right in here, if you look here, you can see what that water or aquaporin will look like. You can see it's very polar, allowing that polar molecule to span through a very hydrophobic cell membrane. Where do we see examples of this? Well, if you want to get technical, if you look at the kidneys, and you have the kidney tubules, and if you go even deeper, you can see the aquaporins that are found in those kidney tubule cells and what they will look like molecularly. You also have glucose transport proteins. So when we think of glycolysis and glucose is coming into the cell, it's going to use a transport protein that is a carrier protein. So glucose will come in. The cytoplasmic of epithelial cells are high in glucose concentration. The blood will be low in glucose concentration. So ultimately what would happen there is you get glucose to pass through. A little bit of this is when you look at glucose being taken into the liver, it is stored as glycogen. It could be converted to pyruvate. It could be converted into a fatty acid, depending on what is needed. But ultimately, the chemical message there would be the insulin binding to the insulin's receptor in that particular cell membrane protein. So those are all the types of passive transport. So passive transport is requiring no energy. The molecules are going to move down their concentration gradient from areas of high concentration to areas of low concentration. There are three types. There is the simple diffusion, there is osmosis, and there is also facilitated diffusion. The reverse of that is active transport, and active transport requires energy, and energy is going to be formed and used in the form of ATP. Basically, you have proteins that are going to transport substances against the concentration gradient. So you're going to be pumping molecules from area of low concentration and moving them into areas where they are already highly concentrated. The best of example of this is the sodium-potassium pump, or when we get into the metabolic pathways, the proton pump. So electrogenic pumps are going to generate voltage across a membrane. We do see this with the sodium and potassium pump. Sodium plus and K plus are ions. So what will happen is we're going to pump sodium ions out of the cell, and when those protein channels open, you're going to bring potassium ions into the cell. We see this with nerve transmission, nerve impulse conduction. So ultimately there you see that sodium is going to be at low concentration inside the cell, inside the cytoplasm. It's going to bind to the carrier protein there to help them get across. What will happen is an ATP molecule will come in. It binds that channel protein, and now we say that the channel protein has been phosphorylated. Ultimately, when the channel protein is phosphorylated, it's going to change its conformation. So when you talk about conformation, you see it undergo a shape change. And those sodium ions are going to be expelled to the outside of the cell. They're going to go extracellular. Now open, this will now allow potassium ions to enter into the cell. Potassium ions will come into the cell. They will bind into the channel protein there. The phosphate group will be released, and ultimately it will allow for the potassium ions to be expelled into the cell then, into the cytoplasm. A proton pump is going to push protons across a membrane. The best example of this is in the metabolic pathway for cellular respiration, where we see it occur in the mitochondria, and we get the production of ATP. So you can see the proton pump is what we refer to as chemiosmosis. We're going to move hydrogen ions across the membrane there. Co-transport is when you have membrane proteins that enables downhill diffusion of one solute to transport or drive an uphill transport of another molecule or solute. So we see this with sucrose and hydrogen ions. Sucrose and hydrogen ion co-transporter would be sugar loading. So if you look here, you can see hydrogen ions are going to pass through their proton pump, a type of active transport there. And ultimately, this will then allow for the diffusion of these molecules. When you're pumping them into areas where they're highly concentrated already, it will allow for the simple diffusion of hydrogen ions to pass through a sucrose hydrogen ion co-transporter protein. And as the hydrogen ion diffuses back across, along with it goes sucrose. So passive versus active transport. In summary, passive transport requires little or no energy. Active transport is going to require ATP. In passive transport, you're moving molecules from areas of where they are highly concentrated into areas of where they are at low concentrations. And it's the reverse for active transport. In active transport, you're moving solutes or molecules from low concentration into areas where they're highly concentrated. In passive transport, because molecules are moving from high to low, they're going down the concentration gradient. In active transport, because you're going from low to high, you're going against the concentration gradient. Examples of passive transport include diffusion, osmosis, and facilitated diffusion. Examples of active transport include pumps and exo and endocytosis. So here you can see a summary picture then of both passive and active transport. Osmoregulation. When we talk about organisms and how they osmoregulate, if you should take marine science, you will learn about some osmoregulation techniques. Basically, you're going to control the solute and water balance within the organism. For some organisms, they have this thing called the contractile vacuole. It's going to be a bilge pump, which is going to force out fresh water as it enters by osmosis. You can see this in paramecium. This very large freshwater protist is paramecium caudatum. And there you can see the contractile vacuole. It's pretty cool to look at under the microscope. It will get very large and all of a sudden it will like close up, forcing fresh water out of the cell that would enter via osmosis. You also have bulk transport, transport of proteins, large sugars called polysaccharides, and other large molecules. Bulk transport, there are two types. One of them is endocytosis, and in endocytosis, you're going to take in macromolecules and form new vesicles. In exocytosis, the vesicles are going to fuse with the cell membrane and expel the contents from inside the cell to the extracellular part of the cell. Two types of endocytosis include phagocytosis, which we call cellular eating, because they're bringing solids into the cell itself. When you bring it in, solid in, we consider that a food vacuole. There's also penocytosis. Penocytosis is what we consider cellular drinking. We're often bringing in fluids. Then there's this idea of receptor-mediated endocytosis. And receptor-mediated endocytosis is when you are going to have ligands bind to specific receptors on the cell surface. And we'll look at that a little bit later. Ligands are going to bind to a receptor protein, which is going to ultimately create a message for that to bring in those substances into a coated vesicle. So how cells talk to one another. Ultimately, this is where we get into looking at some of those extracellular features like the glycoproteins, glycolipids, so on and so forth. So where these carbohydrate chains come into play. So you have these signaling molecules that serve as chemical messengers, allowing cells to communicate with one another. Cell receptors are going to bind to specific signaling molecules that are found on cell membranes. Once the signaling molecule and the cell receptor bind, it creates a cascade of events that occurs that are ultimately going to elicit a cellular response. This is basically what we would consider a single transduction pathway. So you're going to have this message come up to the cell, and it's going to signal the cell to do something. Examples of single transduction pathways, you could talk about the pancreas releasing the hormone insulin, which is transported to the blood vessels in the liver. This will cause the liver to store glucose, which is blood sugar, as glycogen. So when the pancreas releases insulin into the blood, it's going to signal the liver cells to have the uptake of glucose. And remember that the storage form of glucose or carbohydrates in animals is glycogen within our liver. Failure to store glucose as glycogen leads to a medical condition called diabetes. This is, when you think about it, maintaining homeostasis. This is where we get into, we looked at a little bit earlier, negative feedback loop mechanisms. So here you can see an example of cell signaling. Receptors are going to bind to that signaling molecule, becomes activated, and initiates that transduction pathway. So that signaling comes up to the receptor. Ultimately, when it does, it's going to send a message. So this would be the transduction pathway then, which is going to basically require a series of relay proteins that ends when a protein is activated. So these relay proteins are going to carry that message all the way to where it is needed. And ultimately, it's going to trigger a response. And that's going to be the targeted proteins that are going to bring about that cellular response. So targeted proteins can be structural proteins, they can be enzymatic proteins, or they can be gene regulatory proteins. And we'll talk about gene regulation when we get into molecular biology. But ultimately, if you have a structural protein that is going to receive that response, the cellular response could be altered shape or movement of the cell. If it's an enzyme targeted protein, basically the response then would be an altered metabolism or cellular function. And if it's a gene regulatory protein, the ultimate response then would be to have altered gene expression and the types and amount of proteins that are going to be produced. So maybe you're producing so much of a protein, you want to shut that gene off. So you send out that message to inhibit that protein from being made by ultimately rendering that particular gene inactive. So that's it for our chapter on membrane cell structure and membrane structure and function, and looking at some dynamic cell processes. We will revisit some of these topics in lab, and we'll do a little bit of in-class discussion. Thank you very much, and have a nice day.